You know, for almost all of human history, there was an entire universe living and breathing right in front of us that we had no idea even existed. A world of tiny organisms, the complex machinery inside our own cells, all of it was completely hidden. The microscope, well, it changed everything. It made the invisible visible and completely rewrote our understanding of life itself.
But that leads to a really fascinating question. I mean, it's one thing to just make something bigger, but how do you actually see something that's almost completely transparent to begin with? Okay, let's dive into this puzzle.
Okay, take a look at this. These are your own living cheek cells, seen through a pretty standard microscope. And as you can see, they're basically just little see-through bags of water. You can barely even tell they're there. And this right here is the fundamental challenge for biologists and doctors. How are you supposed to study a living process if you can't actually see it?
So, here's our game plan. We're going to break down this invisible problem. Then we'll go way beyond just making things bigger. We'll get into the art of proper lighting, learn some amazing tricks to make things pop, and finally, see how scientists can literally paint cells with light.
First up, let's get right to the heart of the matter. Why is it so incredibly hard to see living things up close?
The whole issue really boils down to this. Biological stuff is mostly transparent. Light just passes right through it. Now, for hundreds of years, the workaround was to stain things with dyes. And that's great if you want to see the structure of something that's already dead. But here's the massive catch. That staining process, it almost always kills the cell. So if your goal is to watch a cell divide in real time, staining is a total non-starter.
Okay, so if we can't stain them, why not just zoom in more? Just crank up the magnification, right? Well, it turns out that making something look bigger doesn't automatically make it any clearer. And that's a really crucial distinction we have to get our heads around.
And that is the difference between magnification and resolution. See, magnification is easy. That's just stretching an image to make it bigger. Resolution, that's the magic. That's all about clarity. It's being able to tell that two things that are super close together are actually two separate things. You can have all the magnification in the world, but if you don't have the resolution, all you get is a bigger blur.
This is a fantastic way to visualize it. This giant thing on the ground is basically an eye chart for spy satellites. Now go ahead and try to zoom in on it with your screen. You can magnify it as much as you want, but at a certain point, those smallest bars just mush together into a gray block. That's not a problem with your screen's magnification. That's the limit of the resolution in the original picture. So the key takeaway here is we need clearer, not just bigger. So how do we get that clarity? You might think it's all about having better lenses, but the secret, it turns out, has just as much to do with the light source itself. How you light up your sample is, well, it's everything. Early microscopes had this really frustrating problem. If you just pointed a light bulb at your sample, you wouldn't just see the sample, you'd also see a perfectly focused image of the hot, glowing, coiled wire from inside the bulb, right on top of everything. The lighting was a mess, it was uneven, and it just ruined the view. The solution is this really elegant system called Kohler illumination, and it was developed more than 100 years ago. It's a clever series of lenses and rings that basically takes the messy light from the bulb and perfects it. It smooths out all the unevenness from that glowing filament. The end result is that your sample is sitting in this field of perfectly uniform, beautiful, bright light. There's a reason it's still the gold standard in pretty much every research microscope today.
And you know, here's a fun fact. The guy who developed a lot of the theory behind this, Ernst Abbe, wasn't just a scientific genius. As brilliant as he was for figuring out the physics of how images are formed, he was also a huge social reformer. We're talking about the 1800s here, and this guy was introducing the 8-hour workday, paid vacations, and even profit sharing for his employees. A total visionary in more ways than one.
Okay, so we've got a sharp image, we've got perfect lighting, but our cells are still transparent. Now we get to the really clever part, using tricks of light to create contrast where there basically isn't any.
So on the left is our old friend, the washed-out cheek cell. But on the right, that is the exact same cell. It's being viewed with a technique called dark field. All you do is put a tiny little disc in the light path to block the direct light. So now the only light that gets to the lens is the light that is scattered off the structures inside the cell. The background goes completely black, and the cell itself just seems to glow. All of a sudden, you can see all this texture.
But we can do even better than that. Here's another comparison. The image on the right was made with a Nobel Prize-winning technique called phase contrast. Here's the wild part. As light passes through different parts of the cell, like the nucleus, it actually slows down by a tiny invisible amount. Phase contrast is this genius optical trick that turns those invisible delays into visible changes in brightness. I mean, look at that detail! You can clearly see the nucleus and all kinds of structures inside.
This invention by Fritz Zernigate was an absolute game-changer. For the very first time, scientists could clearly see the internal machinery of living cells without having to kill and stain them. It just blew the doors wide open for a whole new era of discovery.
So we can see the overall structure of a transparent cell, which is amazing. But what if we want to see something very, very specific? What if we want to track, say, just one single type of protein as it moves around? For that, we've got to go one step further and learn how to paint with light. This brings us to the world of fluorescence microscopy. The basic idea is you attach special fluorescent molecules, think of them like tiny targeted highlighters, to the specific things you want to see. Then, you shine one color of light onto the cell, and only the parts you've labeled will absorb it and then glow back at you in a completely different color.
And the results are just breathtaking. I mean, look at this. This is a cancer cell where the nucleus has been labeled to glow blue and the cell's internal skeleton is glowing green. We're not just seeing the outline of the cell anymore. We are seeing specific functional parts in brilliant color against a pure black background.
Here's another one. The cell's skeleton is labeled in green here, but the fluid and all the little transport bubbles inside are glowing magenta. It's like we're looking at the cell's internal highway system and all the cargo that's moving around inside of it. It's just astonishing to see. And we can use a bunch of colors at once to see how different parts of the cell relate to each other. This is a tiny little bubble made from a cell membrane. One type of molecule is tagged green, another is tagged magenta. And you can immediately see that the membrane isn't just a uniform blob, it's actually organized into distinct neighborhoods. This is molecular biology made visible. So, let's just trace our journey back. We started with this huge problem. Living cells are invisible. We learned that the answer wasn't just making things bigger, but getting crystal clear resolution. Then we mastered the lighting and used these incredible tricks like dark field and phase contrast to make the invisible pop. And the result? A spectacular ability to see the building blocks of life doing their thing in real time. We've gone from a blurry, indistinct blob to a vibrant, multicolored, living machine. Our ability to see has been completely defined by how clever we can be with light. It really makes you wonder, doesn't it? What other secrets are still hiding in the invisible world, just waiting for the next brilliant trick of light to finally bring them into focus?